Can you see my screen now?
Yes.
Okay. So this is week 6 TA session, and as usual, we'll solve some week 5 assignments that you have already submitted and do some sample problems for week 6. So the topics covered in week 5 were aggregation of proteins, kinetics of protein aggregation, and effect of surfaces on the aggregation of protein. And for week 6, host response to biomaterials, cell adhesion, and biocompatibility of the biomaterials was covered. So we'll first start with week 5 assignment problems. So this was the first question. Which of the following sequence represents the event of protein aggregation? Did you answer this only? Native, unfolded, oligomer, and aggregate?
Yes.
Yeah. So this is the correct answer. The usually the protein would be in native state, but due to some external factors, it will get converted into unfolded, and then it will form oligomers and it will become an aggregate. Now the next question was aggregate results in formation of the following secondary structure. So during the transformation of these proteins to from a naive state to misfolded or um, misfolded state, it involves transformation of the alpha helical structures to beta sheets. So the correct answer here would be beta sheets. These beta sheets then interact further and form aggregates. Which of the following diseases arise due to protein aggregation? So the options given were Alzheimer, Parkinson, prion, and all of these. Correct answer would be all of these. So the Parkinson and Alzheimer's are basically neurodegenerative diseases that occurs due to misfolding of proteins, and prion is another such disease wherein prion is basically a protein that can trigger normal proteins to in the brain to fold abnormally, and it can affect both humans and animals. And the most common form of the prion disease is Creutzfeldt-Jakob disease. So here the correct answer would be all of these. The next question was which of the following methods can be used for characterization of protein aggregation? And the options were thioflavin T fluorescence, morphological analysis, residual protein analysis, and all of this. Again, the correct answer would be all of these. So thioflavin T can be used. This basically detects the beta sheets, and more beta sheets are uh, the alpha helices are converted to beta sheets, and that leads to protein aggregation. So more the beta sheets, more protein aggregation. That is how it is related. So thioflavin D basically detects these beta sheets in the protein aggregates. And then morphological and the residual protein analysis will basically show the remaining bulk concentration of the protein that decreases with time. And morphological analysis, you can do AFM or AFM like and microscopy. So uh, in microscopy, you'll basically get to know uh, these protein changes in the protein structures that consist of the morphological analysis. So here the correct answer would be all of this. Now next question was which of the following equation is applied in dynamic light scattering method? So here Stokes Einstein equation would be the correct answer. So dynamic light scattering is basically a laser beam has passed through and the fluctuations of the scattered light are detected. And the Stokes-Einstein equation, that is dH equals to kBT by 3 pi eta d, where kB is Boltzmann constant, T is the absolute temperature in Kelvin, eta is the viscosity, and D is the diffusion coefficient. So this gives a diameter dH, and based on that, uh, we can also analyze the protein aggregation. Next, this was the question: which of the following, which of the intermolecular force is or are involved in intermolecular interactions? Again, all of these would be the correct answer. So the dispersion forces, that includes London and van der Waals forces. The electric double layer, that is when material is exposed to the body fluid and initial proteins get attracted to this material, and then one more round of proteins will form the second layer. So there is a electrical the solutes, sorry, the solutes will the charges of the solutes will come and coat the material. So these charges would form electrical double layer. And the short range forces, such as the hydrophobic bonding, hydrogen bonding, and ion pairing and dipole interactions, are also involved in the intermolecular interactions. Now, this was the next question. What is the first step of protein aggregation on solid surfaces? So, here the correct answer would be adsorption. First, adsorption takes place, and then the next steps of unfolding and aggregate formation will take place. Sakshi, I hope you're still with me.
Yes, yes, I'm following.
Okay. So, this was the next question. Which of the event occur during the lag phase of protein aggregation? So here the correct answer is conformational change. So after experiencing external factors, the native protein undergoes conformational change. Then it forms nuclei and formation of aggregates happens. So the correct answer would be conformational change. And this is the next question. What is the effect of the initial surface roughness on protein adsorption? So here the correct answer would be increases. So the initial surface roughness increases the protein adsorption. Uh, this graph also shows wherein the experimental results, where in one of the experiments, this is what it was observed, that the more the surface roughness, the more adsorbed mass was there. And also, if you see it like this, the more the smooth surface, it will give less chance for the proteins to interact with the material than a slightly rougher surface. So more surface roughness will increase the protein adsorption. And this was the next question. Which of the followings will cause protein aggregation? The options were upregulation of chaperones, interference with post-translational peptide changes, inhibition of aggregation. None of these. Uh, Sakshi, what did you answer for this question?
I answered none of these, but it seems that
Right.
upregulation of fat. But then in the
Yeah,
video,
I wanted to say that you guys can put in the discussion forum saying that the answer would be none of these, because even I think it would be none of this, because upregulation of molecular chaperones will basically convert the unfolded into more folded proteins or native proteins, right? As it was taught in this slide.
so how do I have to put it in the disc? Like from where?
Uh, I'm not really sure, but I had I had I think there is a discussion forum where you can mail or put in that forum saying that the correct answer for this would be none of these. And you can also tag the lecture there in saying that in this lecture it was taught like this.
Okay, okay.
Okay. And I have some sample questions for week 6 assignment. So for week 6, this is the first question. Which of these is the correct sequence for homeostasis? Can you answer, Sakshi? I'll give you some time to go through this question. So the steps are formation of fibrin plaque, vascular spasm, activation of coagulation cascade, and then formation of platelet plug. But what will be the correct sequence?
Fourth option.
Fourth option, formation of fibrin plug, then activation of coagulation cascade. Mm, okay. No, the correct answer would be third option. So first vascular spasm will be there near the tissue injury, and then that will be followed by formation of a platelet plug, and then eventually it will activate the coagulation cascade, and lastly there will be a formation of fibrin plug. So the correct answer would be the option 3. Okay, I have the slide as well. So yeah, vascular spasm will lead to constriction of the blood vessels, and then more and more that will lead all the platelets to the side, and the platelets will form a plug, and then that will trigger the coagulation cascade, and more clotting factors will form the fibrin clot, the final fibrin plug actually, and then eventually that will clog the that is how healing process happens. And this was the next question: where do all pathways of complement activation converge? C4 and C5 convertase, C2 and C3 convertase, C3 and C5 convertase, or C2 and C4 convertase. Do you want to answer, Sakshi?
I don't know this.
Okay, so the correct answer here would be again option 3, C3 and C5 convertases. So if you go through the lectures, here it shows the classical pathway, the lectin pathway, and the alternate pathway. All of them converge near the C3 and C5 convertases. And this is the next question. Which of the following is not true about integrins? The options are: they are transmembrane receptors, they mediate attachment of cells, they are important in cell signaling, and they are part of the ECM.
Fourth one.
Yes, correct. So they are not part of the ECM, but they help in mediating attachment of the cells to the ECM, and they are transmembrane receptors, and they also help in cell signaling. So fourth is the correct answer. Okay, these were all the questions I had today. And if you have any doubt, we can go through that. Or else, if you want me to repeat any other question.
I don't have any doubts.
Okay, then let's meet next week again with week 6 assignment, solved assignment.
Yes.
Okay.
Thank you.
Thank you for joining.